Okay, so we'll start the next session. So we're delighted to have Jim DiCarlo give the next tutorial. So Jim is a professor of neuroscience at MIT. He's in fact the head of the Brain Cognitive Science Department and an investigator at the McGovern Institute for Brain Research. Jim.
Okay. Oh, thank you. So I want to thank the organizers for giving me a chance to speak to all of you. Is everybody hearing me okay? Yeah, okay. So great, thank you. So I'm going to I know this is a tutorial and 2 hours is a long time, so my plan is to give you guys a bit of an introduction to who I am, what my lab does. I know many of you may not know me. And then I'm going to break the talk into 3 different parts, and all of them are going to be doing comparisons between things like behavior in humans or monkeys and neurons and machines, which I know many of you are interested in and I think is in the spirit of this conference. Okay, so let me start. You know, the problem that we are interested in is the problem of visual object recognition. I know many of you are interested in that as well. This is a deep problem in human cognition, and it's of central importance. This is just to remind you that the representations of objects in the brain we think of as a substrate for higher cognition, and this is why this is an important problem. They are the things that get laid down in memory, that we make decisions about in the world and guide our actions. I don't think I need to tell this audience that. And of course, if you're an engineer, many kind of tasks rely on accurate identification and categorization of objects. As engineers ourselves, we like to think of this problem, even though we're a neuroscience lab, we like to focus on what is hard about the problem, and success would be being able to implement in algorithms things that have successfully solved the problem. That's what we view as success, I think, like many of you. I put this up to sort of remind you there's some areas where machines are already better than us. There are other areas that we think they're close but not quite better than us. And so object recognition, I think we're currently better at, depending on how you define it, although things are getting close, as you'll see. The goal of my lab is really to try to discover how the brain solves this problem. And again, I mean in the sense of algorithms, so that we would be effectively, if we have that understanding, then this should mean that we could move this over there, because if we understand it, we should be able to build it. And I'd also like to say, for those of you who think about the brain, the problem of object recognition in the ventral stream as a hierarchical structure is really a gateway problem to more deeper understanding of cortical processing more broadly, not just for vision, but perhaps for other senses and even cognition. Okay, so by way of introduction for object recognition, this is the way that computer vision folks often view the problem, what they would call detection, and it looks something like this, putting bounding boxes and labels around things in the scene. And it's often sort of assumed that humans have sort of infinite ability to do this, or somehow we're really great at this. And we are great at this, as I'll show you, but I want to set some context here to tell you how we actually do it. So object recognition as solved by primates is not instantaneous, do all that labeling. What you really do, the part of your brain that's really digesting the object called the ventral stream that I'll show you about in a moment, it really looks at about the central 10 degrees of the visual field. That's where you have high acuity vision. If you hold out 2 hands at arm's length, the width of 2 hands, that's about 10 degrees of visual field. And so for many of you in the room, maybe about halfway through this room, this is about 10 degrees. And so what I want to remind you, so you're not really digesting the whole scene at once. There are other parts of the brain that tell you sort of, hey, there's other stuff over here, you might want to move your eyes. But that's the other thing that your brain does, is it samples the scene. So it fixates, usually for a period of around 200 to 500 milliseconds, for just so a short period of time. And then you're not really aware of this, but you're making rapid eye movements called saccades, followed by fixation, saccade fixation, as you explore the interesting points in the scene. I'm bringing this up to get you to a reduced form of the problem of not just this full scene understanding, but so that what your ventral stream engine, as I call it, is taking in is something that looks a little more like this, if you watch this little movie here. So that's about 6 or 7 clips from that saccadic sequence that I showed you. And you might have noticed if you watched that you could recognize one or more objects in each and every one of that central 10 degrees scene. So that reduced form of the problem is how your brain is sort of digesting and then moving and digesting. And it's that reduced form of the problem that we call core object recognition, the central 10 degrees of visual field, and brief viewing times on the order of 200 milliseconds or so. That's your natural saccadic fixation duration. That's what we call core object recognition. That's the phenomena of interest to us, and we think it is the gateway problem to understand, to really build a foundation for general scene understanding. So you're really good at this problem of core recognition, as you noticed in that last little clip, but I like to also show this movie. This is called Rapid Serial Visual Presentation. This illustrates the same idea, but not from a coherent scene. Now you're noticing that you can probably recognize one or more objects in each and every one of these images, even though they're coming by quite rapidly. This is about 6 frames per second. So on the order of 150 milliseconds or so, you can easily sort of say at least one identity out there. And I didn't have to tell you what kind of objects to expect, like Star Wars characters. I didn't have to tell you, probably many of you saw Leaning Tower of Pisa. There are many things you can quickly map to memory, thousands of objects that I didn't have to pre-cue you. So you don't need sort of strong forms of attention to do this task. And you're highly tolerant to variation, so the ability to recognize Yoda independent of the viewing conditions. And that, as many of you know, is the crux computational problem. Okay, so now that I've reduced the problem a little bit for you, now I want to tell you about our model system. We'd, of course, like to understand how this brain solves the problem. This is the human brain. We work primarily on this brain. This is the non-human primate, the rhesus monkey. This is the lateral view of the rhesus monkey brain. It's not shown to scale, but you see it has grossly the same layout as the human brain. And the reason we like the rhesus monkey brain is that we know the parts of the brain that house the critical circuits for solving visual object recognition. That's the series of areas called the ventral visual stream. It starts in the back of the brain in an area called V1, primary visual cortex, through V2, V4, and on to this area called infratemporal cortex, or IT. And I'll spend a lot of my talk talking about IT here. So we think we know where these representation lives from a number of studies, for instance, lesion studies of this part of the brain in monkeys. These neural representations are projected to regions of the brain involved in decision and action and to regions of the brain involved in memory. So again, the substrate of higher cognition. So we can study these, unlike the humans, we can study these in the monkeys at a level of abstraction that we think is more appropriate. Rather than, say, voxels in an MR scanner, we can study the spiking activity of neurons at every level of this system here. And I'll show you some of those data today. And because these are monkeys and not, say, a mouse, we can really directly compare the abilities of this creature with that creature, and they're actually quite well matched. And even when you do things like look for parts of the brain using fMRI, you can do that in humans, and you find a lot of homologies in the monkey. So this system is close to the human, but is more approachable, much more approachable than the human system. Okay, so the ventral visual stream of the monkey, as engineers, we like to lay it out like this. So as this convoluted cortex here, it has millions of neurons in each of these visual areas. So on the order of 100 million neurons in V1. And you see these are roughly drawn to scale. It gets a little less neurons in each visual area. These are meant to symbolize neurons. There are sort of feed-forward anatomical pathways, feedback anatomical pathways, and also intracortical connections. So we consider each of these areas to be what you would call a new population representation, a new representation of the image. And part of the reason that we and neuroscientists believe this is because each area contains a complete retinotopic map of the visual field. That is, each point samples a different portion of the visual field. So it covers the visual field. Here it's the full visual field. By the time you get down here, again, it's around more of that central 10 degrees. Okay, so you have a retinotopic map, another retinotopic map, another. So you're changing features somehow, and that's the interesting computation going on in here. In IT, it's less clear because you're more feature-oriented. There's much less of a retinotopic map when you get to IT. It's not even clear if it's a single area or not. Okay, so here's a sense of what we think is going on when you do that task that I showed you earlier, core recognition, is that when you show an image like this, it makes your retina kind of captures a nice image of it, like a nice digital camera. That is rapidly transmitted in about 100 milliseconds through these series of visual areas with a latency of about 10 milliseconds between each area to a new pattern of activity in IT cortex symbolized here by these red dots. So this is the activation pattern of a population of neurons in IT cortex. It's no longer a photograph. You have an isomorphic representation here, a photograph, and here you have some other new pattern, some new feature space that's produced. And again, the time lag is about 100 milliseconds here. And when you were watching this movie, a new pattern, a new image, new pattern, and when you were watching this movie here, your homolog of IT is clicking along of a lag. For you, it's a little bit longer, closer to 150 milliseconds following these images. And it can easily follow at this pace here, and we think that's what explains your ability to easily be able to do the task at this speed of presentation. Okay, so that's a bit of background. We think there's key cortical circuits that live here. We'd really like to understand what's going on here, as I said in my first couple of slides, and that has remarkable potential, maybe not just for vision, but also for other sensory systems. So just to orient you to the kinds of things that you can do in a systems neuroscience lab is that we often spend a lot of our time doing what's called psychophysics, so that's the study of behavior in monkeys and humans, neurophysiology, which is the extracellular recording of neural activity in different levels here, as I mentioned, computational models that I'll show you some of today. You can do functional imaging, I mentioned, on both humans and monkeys to map one to another. And you can do things now in monkeys where you can intervene. So with tools like light, you can turn on and off neurons here so you can start to interact with the system to test ideas. So that's sort of the rough scope of our toolbox here. And I won't tell you about all of those today, but I'll touch on some of them. So the idea, again, is you have this 1 million dimensional image space here that's somehow transformed to something that's somewhat less than 10 million. And I say 10 million because that's the number of output neurons roughly in IT cortex. And it's probably well less than that, but that's an upper bound. Okay, so first I need to ask, or to introduce to you, why do you even need to have this system here? Maybe this audience sort of already knows that, but I need to often remind some audiences of why you need to transform the image here from the pixels. And the answer is, of course, that it's very challenging to do recognition on the image itself because each object, say this car, can produce an essential infinite number of different images due to variation in, say, position, size, pose, illumination, background clutter, occluding clutter. You can have interclass variation. You still might want to call all these a car. All of this together is the ability to tolerate this, what's called identity-preserving image variation, is called invariant object recognition. And so what you're seeking are to extract these latent variables of the world, in this case the latent variable of identity, and that's hard to do at the pixel level. And I like to think about this from a geometric point of view. I think others like this view as well, but if you haven't seen it, it helps me a lot that you think of what's happening in a population representation is that each neuron here in this each axis here is the firing rate of a neuron. Think of this population of retinal ganglion cells like a camera. And so when you had a particular image of this one particular person, let's call him Joe, he's one point in this high dimensional space, million dimensional space. And if Joe undergoes a transformation like a change in pose, he sweeps out a new set of points, and this is a highly curved set of points in this original space. And another change in pose, so that's 2 degrees of freedom right here. You can imagine all the degrees of freedom that I showed you with that car example, and they sweep out a very small set, thin set of points that are in this ambient space here. Those are all the potential points that Joe could give rise to, and that's called Joe's identity manifold. So it's a manifold of points. Now, the reason I like to introduce this is because now once you think about these manifolds and the induction of these manifolds from these other nuisance parameters like changes in position and scale, that now you can start to see what the problem is that what you really need to do is produce a population representation that we call an explicit representation, one that is easily decoded. So this is just called a good representation. So an explicit representation of shape, we operationally define it as one where you can put in separating hyperplanes, that you can easily find linear decoders that can say this part of the space corresponds to images of Joe, and this corresponds to images of, say, not Joe. And I need to remind my neuroscience colleagues that a linear classifier here is really an approximation of a dot product of something a downstream neuron can easily do. So it's not a complicated operation. So we don't view this as a sort of magic box. It's producing the good representation is the challenging part. And we also want to point out for this audience that you want to be able to find this with relatively low numbers of training examples here. So it's not just being separable, but it's being able to do it with low number of training examples. And I want to also point out that we think of the representation in IT cortex as probably more of a shape representation. It's not necessarily an identity or saying that there's a semantic label like a car, but it easily can support a semantic label like a car with this kind of operation. Okay, and I presented all this as sort of manifolds, but this idea right here of testing the explicitness of a representation with these kind of linear classifiers does not require that you believe that everything's manifolds, because of course some transformations are not going to be clean like changes in pose here. Okay, so what you have, the reason I've introduced this is you have these tangled manifolds at the original image here on the retinal space, and that makes it very, very hard to solve this problem, at least with low numbers of training examples. When you go to these high visual areas, we think the situation is much more like this, where you have flattened or untangled manifolds, so that you can easily put a separating hyperplane, for instance, to separate Sam in this case from other things, or Joe from other things. And you've also noticed here, you don't throw away, I mentioned some other things that I call them nuisance parameters, but there are other, you keep some of that information. So for instance, there's still information about position and scale here that you can also still extract. So you've just made a nicer representation of the latent parameters, we think. Okay, we call that an explicit or untangled object information. In the language of computer vision, this is a poor set of visual features, and this is a powerful set of visual features. So these are all just different ways of saying the same thing. Okay, so this was all introduction to sort of remind you of why the brain needs to transform the pixel image from here to there. And now I'm going to sort of just give you an overview of what I'm going to try to tell you today. So that was the general introduction to what my lab works on. Here's what I'm going to try to tell this audience today. So I'm going to make some comparisons. First, I'm going to try to compare monkey neurons and human behavior. And this comparison is going to suggest that IT population codes are indeed one simple step away from matching object recognition behavior in humans and monkeys. Then I'm going to compare machines, and I'm going to show you a particular model that we built with monkey neurons. And this suggests that building machines that have a behavioral goal of good recognition performance actually starts to lead to an understanding of what these neurons are doing. And then in the last part of the talk, the shortest part, I'm going to talk about a comparison of some of the machines you heard about earlier this morning if you went to Rob's tutorial versus monkey neurons and human behavior. And he already introduced some of that, so I will go over that in a little more detail if we have time. And what I'm going to try to do is stop in each of the end of these sections so that you guys have time to ask questions, because I know this is meant to be a time for you to, it's a tutorial. So before I get into it, maybe I can stop right here and see if there are any sort of burning issues that people want to raise or questions at this point. Okay. All right. So let's go on. So monkey neurons versus human behavior. So we're going to make this comparison now. So this story starts with thinking about, okay, you're doing a transformation here, but there's this question of, I've sort of implied that there's a solution up here to recognition in those introductions I gave you. So I've said that the solution lives in IT and that it's sufficient in some ways. That's what I sort of showed you schematically. But this has been an idea in the literature for a long time, but it hasn't really been shown. So it needs to be sufficient in terms of performance. And if it's really the thing that the brain is leaning on, it should kind of make the same patterns of mistakes that the human makes. At least it should show the same patterns of difficulty that the human has across different types of tasks or different types of images. So that's what we're going to test here. And we're doing that because if we can find neural codes in here, then that establishes a more quantitative link, not just a conceptual idea, but showing here is exactly how you can read out of, say, these neurons here to support the behavior of interest. Okay. So now I get to introduce you to the idea of extracellular physiology. So when you go in and record from neurons and IT cortex in a monkey, this is some old work that I did a while back with my colleague Tommy Poggio at MIT and a couple of postdocs, Gabriel Kreiman and Xiao Heng. We record from IT neurons. This was with single electrode recording. So there's a single electrode. You move it down. You get it close to a neuron extracellularly. You can listen to its spiking activity. And when you plot it, it looks like this. Each tick mark is the firing of a neuron, an action potential out of the neuron. You can barely see those little dots. Each row is a different presentation of the same image. You see 5 images presented here. Typically, when we present these, they're randomly shuffled, and then we just resort things so that you can see here. So even though the neurons are sort of noisy and that they don't, that you don't see the exact same pattern of spikes, you can see on average a tendency to prefer this image and this image over, say, these. You see a lot more tick marks here than over here. This is at one neural recording site. If you move the electrode and go to another site, you might find a different pattern of image selectivity. So now, for some reason, it likes these images. Here you go over here and it likes these images. Nobody really knows yet what makes these neurons go, but you can easily get this kind of phenomena out of IT cortex. And so I should point out, this is in an awake fixating monkey. The monkey is just fixating while his images are flashed up in random order, typically for a period of about 100 milliseconds. Again, that's after that core recognition idea to just keep this short duration, similar to your fixation duration and the monkey's fixation duration. And what Tommy and I showed with Gabriel and Xiao a while back was that you can take this population activity, and so this is a schematic of a bunch of different neurons now recorded out of IT, and we would do things like compute codes, or what we call codes out of this activity. For instance, a simple code is just count the spikes in a 100 millisecond time window locked to the stimulus onset. Count all the spikes in there, and so you get one number for each neuron. So you say you have n neurons, you have n numbers. So this is a feature vector, if you will. It's a point in an n-dimensional space where each axis is the activity of one of these neurons on this presentation of this one image on one trial. And so now you can imagine other presentations of the same image or presentations of different images. And now you have this sort of neural population or neural state space that I introduced you to a moment ago. And now you can ask, how easy is it to put decoders on this space to say, what is this object out here? For instance, is it a face or is it not a face? You can ask, how well can a human face detector do on this kind of population activity? And you can, of course, use tools like SVMs or simpler classifiers like correlation classifiers to go ahead and test this. And Tommy and I did this for a number of simple objects. This is way back a long time ago just to show proof of idea. And it turned out in that case, you didn't need that many sites to do what was really good performance. I mean, I'll put good here with the star because this isn't benchmarked in any way. But you didn't need that many sites to get good real-time performance. And what was most important to show is that if you train a decoder to say at one position and size, that it would generalize quite well, not much drop in performance to new positions and size. So the representation, when you learn from it with few training examples, would automatically have properties that it would nicely generalize to at least changes in position and size. And you couldn't do that kind of thing when you compared with early visual cortex. So these were sort of early clues that we'd already thought, based on the work of others, that IT could support this, but this was the beginning of us trying to move to use modern tools to map it more quantitatively to behavior. So now let's sort of ask the more quantitative question that I set up is, how well do these neural codes actually quantitatively support the behavior in terms of pattern of difficulty and magnitude of performance? So I need to introduce you, for the non-neuroscientists in the audience, introduce you to the idea. You may already like the idea that I've set out here that, well, these automatically evoked, and when I mean automatically evoked, it's a passively viewing monkey, images presented, spikes come out of the brain, we record them. Automatically evoked spike rate codes distributed over non-human primate cortex can fully explain, I mean quantitative explain, human object recognition. That's sort of what I've been implying, but that's actually a sort of a provocative statement because there's a lot of other things that you could imagine going on that might be more complex, maybe that makes them more attractive, but certainly there are other things that are raised. So perhaps you might believe, DeCarlo doesn't know what he's talking about. IT isn't going to support object recognition. There's some other part of the brain like V4, prefrontal cortex, LIP. Whatever your favorite brain area is, that could be the answer. Maybe the rate codes in IT, just counting spikes in a window isn't sufficient. You need to look at the patterns of spikes in some detailed way. Maybe that the automatically evoked spike patterns are not sufficient. Maybe you need to kind of get the monkey doing a task, or else the neurons are doing something different, so dramatically different that it's not going to match. Maybe you have to pay attention to exactly which neurons you're recording from in IT, that you need to consider the compartments within IT. And there is evidence that IT is not a homogeneous mixture of neurons. There is clustering of different types of neurons, for instance, with preferences for faces. And maybe you could just say, well, look, this is a monkey. You're saying you're explaining human perception. How could a monkey explain human perception? Monkeys don't know what a chair is or what a car is. But remember, what I'm talking about is a basis to support a semantic label. And we think monkeys do know what shapes are. So you can imagine other things. But I just wanted to say there's other possible alternatives here. But what we set about to do, and when I say we, I mean Najib Majaj, a postdoc, Ha Hong, a graduate student, he's here at the conference, and Ethan Solomon, an undergraduate, with support of many others, we set about to just test this simple idea. And so even though it sounds like a simple idea, it's some work to actually test it, and I'll take you through how we tested it. So what we're going to be doing is comparing populations of IT neurons to the behavior, in this case, behavior of humans. Okay. And so first to do that, you first need to define a set of what we'll call challenging object recognition tasks. Then you need to measure the behavioral performance of humans in all of those tasks. Then you need to go take the same images and measure all the population spiking responses out of IT cortex, measure a sample of them. Then you need to apply the decoders that I mentioned to compute the predicted behavior from that population. Then you can ask, does this prediction quantitatively match this observation here? Okay, so it's a pretty simple comparison, but making these quantitative comparisons is what this talk is about, and I think how we make progress in this field. These are strong correlational methods. They're not causal methods. They're just asking, could this support this based on a correlational argument, not a causality argument. And our goal, but I want to also point out, the goal of our work is not to just say, hey, there's information down here in the brain, but it's to test an idea that this way of reading this information, this particular way, what I'm calling a code, might be the explanation of how your perceptions or your behavioral reports actually arise. Okay, so we're going to define a set of challenging object recognition tasks. Remember, I said the reason it's hard is because you have to deal with all this variation. So that's the kind of challenges we wanted to introduce. We knew this would make it hard for computer vision. So rather than going out and doing ImageNet, we can't show 1 million images to the monkey. We said we're going to do this in a way that is controlled but still hard. We took 3D models, we render them, we control the latent parameters of identity, position, size, and pose. We can then place that on backgrounds. We can put it on a correlated background. We just placed them on uncorrelated natural backgrounds. And the reason we did this is because our work in computer vision was suggesting that a lot of computer vision systems were cheating on the background, even though we knew humans could see right through this. They didn't need the background to know that that was a car. So you get the kind of images here that are not fully natural. They're semi-natural. They kind of have naturalistic statistics. They seem very weird, right? But what you'll see is these kind of really computer vision systems at the time, at least, could not deal with these. So it was a good test in that sense, yet humans could easily do these kind of things. Not for everything, and I'll show you they don't do perfectly in everything. But you could easily, I'll give you an example yourself. So you have these we had full parametric control. I've already mentioned all this. They're not fully natural, but they're challenging for computer vision, doable by humans. Okay, we grouped these into kind of different types of tasks because we wanted to test things that were not only basic level categorization, like is it a face versus not, is it a chair versus not, but also discriminating, say, different types of cars or different types of faces. So more what are called subordinate tasks. And so to give you a sense of this, here you can just watch this. I won't even tell you what the objects are, but just you'll see it's not that hard, but you're not. Okay, so that was about 100 milliseconds. That's what we typically show. So anybody know what that was? Car, right? Okay. Okay, good. Somebody got a plane. A little harder, right? Yeah. Okay, so they're weird, but you can get most of them. You're not perfect. Okay, computers have a hard time with that. And so that's the kind of tests that we've been doing. Again, under this idea of core recognition, keeping those images short, shown at about 10 degrees central visual field. So again, we measure human performance in a controlled way. We did this in both in the lab and on Mechanical Turk. I just want to give you a few details here. They had done this, they were doing this in a blocked mode of either a categorization task, these two subordinate tasks. When we talk about tasks, we break things down into a binary task where you might be saying a car versus not a car. That's not what the humans were doing, but that's how we analyze the data. So we end up with what we started with 144 binary tasks. Later, we reduced these to only 64 tasks. So you'll hear me talk about these 64 tasks, what I mean by binary tasks, throughout the rest of this section. And again, the images were 8 degrees circular aperture at the center of gaze for 100 milliseconds. And this is, again, this idea of core recognition. Here are the data from the human subjects. This is 39 human subjects. This is in over 144 binary tasks. Each color indicates the performance level. This is in D prime units. So high D prime means very good performance. This is what psychophysicists like to use to characterize a performance. It's a measure of sensitivity, and it takes into account bias in any judgments. And what you want to see here is that red means very good. A D prime of 5 means basically 100% correct. A D prime of 0, it's 50% correct. And what you should see here is that humans are not D primes of 5 everywhere. They have a range of performance, what we call a mosaic of performance. But this is a very reproducible pattern. Let me just show you a few examples here to give you a sense. As you might imagine, doing basic level tasks are easier than doing subordinate level tasks. And you can kind of see that in the color scale here. So here's a task that's a low variation. So what's on this axis, we vary the amount of variation. Remember, we had those other latent parameters like position and scale. And so here's a low variation face detection task, face versus these other things. Here's an example of the images. Of course, you see these one at a time, but here's effectively the discrimination you're trying to make. Here's examples of faces. See, they're all frontal facing, about the same size. There are actually different faces in here and different backgrounds still. There's some variation in the images, but you can see it's quite easy to say this is a face and these are not faces. Then if you kind of increase the variation to high, then you get situations like this. So you have these viewing parameters changing. These are faces, these are not faces. Your performance drops, as you saw on the last slide, but it's still pretty high. A D prime here of roughly 3 or 4, which is sort of on the orders of 80 to 90% correct. Okay, so now you go to a different task. Okay, here's another one. Here's a subordinate car task just to give you a feel. Here's cars versus, this is like a beetle versus other types of cars under a medium level of variation. Okay, so you can kind of get a sense that, look, you're not going to be perfect at this. You go to high variation tasks with fine discriminability. You can't do that quickly in 100 milliseconds. You need longer time to do it, or you may not, you may have a very hard time to do that. Okay, so the pattern of performance is reliable across human observers, and that we can use to our advantage, and that's the lever we're going to use here. So to show you that it's reliable, here's the pooled population D prime, so that's these D primes here now plotted, and here's one individual subject. So you can see that each individual correlates well with the average, but there's some variation here, and individuals are not copies of each other, but you can see there's a very strong correlation, about 0.93. And here's an example. So each dot is an individual task. So there's 64 tasks, and that's what I'll be talking about for the rest of this section. So again, we can use this pattern of performance as a tool to ask, is a neural code sufficient to explain this pattern of performance, both in terms of the pattern and its amplitude? And again, this pattern, as I'll show you, is not explained by simple artificial visual representation. Okay, so now we got to go measure some neural data. And the way we do that in the monkeys is we go, in this case, recording with what are called chronic recording arrays. These recording arrays have about 100 electrodes each. We implant typically 3 of them along the ventral stream, typically 2 in IT cortex and 1 in its input area called V4. So these are schematics of places that we tend to put these, and here's a picture of the brain during surgery when we actually put these in. Then we close everything back up, and then the monkey just wears a connector on his head, and we can plug the monkey in every day and record neural activity. Over many, many days, you have often stable recordings, sometimes for months, and then you can collect many thousands of images from these sites on the order of hundreds of multi-unit recording sites here. So this is only a sample of IT cortex, and that's something I'll return to in a moment. The neural selectivity properties are very comparable to those single extracellular electrodes that you saw me mention earlier. And we take several monkeys, so we pool data from several monkeys to build up a larger sample. Okay, these, I'm going to skip through that. This is just a technical point that this allows us to collect a whole lot more data. Let me jump ahead here. So this is to give you a feel for what's going on. This is just schematic. So the monkey's fixating. Images are being flashed at about 100 milliseconds each, randomly sampled, and we're recording from many of these arrays. This is a schematic of one array, so you imagine neurons turning off and on while you're recording this. So and here's an example data I'm going to show you from one channel. So now we're back to these little tick marks that I showed you at the beginning. Those little black dots are tick marks. These are different images. You can see different rows there. Those are different presentations, so what we call repetitions of the same image. So there's 8 images there. We had tested thousands of images. This is just to give you an example. Then you can count spikes in a window. That's a window we're going to vary, just as I showed you earlier. That's, say, a 100 millisecond count window. And here's the count in that window now averaged over 50 repetitions. So that's the mean response to that image averaged over repetitions. And you see some images drive this site more than other images. That's only 8 images there. Here's examples of other categories that were being shown in their images. And we had, again, many unique images from each category. And you see the patterns of response here recorded from this individual. This is one IT site. And as I mentioned, there were hundreds of IT sites. So this is now recording, that's like that one example site. But we could record up to 256 simultaneously. Not all sites were visually driven, and, but we could record up to 256. Okay, so here's what you get out of this. Here's 168 visually driven IT sites. This is a vector response. You may barely be able to see those little colors there. This is that response vector I introduced you to a little bit back in the talk. This is a response to this image constructed in the way I just showed you for those 168 neurons. Remember, we had many of images. Here's a bunch of other images. And in fact, we collected thousands of images. So here's 2,560 images, and now just to give you a sense of the volume of the data. So you have this sort of big mass of data that we've collected. Now we've measured it. These are the same images we had tested humans on. Now we can compute the predicted behavior from these images. We can ask the decoders, how well do you do IT or V4, which we also collected on these kind of images. So how do we do that? So we need to build a decoder that predicts performance on all the tasks. So we need to predict one D prime value for each of those 64 tasks. And what we do is we say we're going to make a hypothesis that a linear classifier decoder, for instance, learn the weights with some fixed number of training examples and then test its performance on new images on that particular task. We build one decoder per class, and we just say, do your best given what I just laid out there. Fixed number of training examples, fix the type of classifier, gets to see the same data, and say, here's you trying to build, this is a hypothetical downstream neuron saying, I want to be a classifier for, say, car versus not car. So you build up a bunch of these decoders. Again, these are simple and specific instantiated hypotheses of how the neural activity could support the behavior that we're measuring. So now you have this. This is just to give you a feel for that. You have many face images. This is 4 examples, non-faces, and what the decoder is essentially doing is finding a hyperplane, projecting on the normal to that plane. You see here, this is if the activity the vectors are well separated when they project. That this would be a case where the performance is very good. This is well separated, could be easily classified. This would have a high D prime, say like 4 or so in this example. For other tasks, say car, not car, you might be some overlap here. The D prime is going to be less than that. Say maybe this might be a case of a D prime of about 2. So you get the idea for each task, even though using the same decoder procedure, you're going to get a different predicted D prime. So now we have this pattern of D primes, and we can compare them directly to the pattern that we measured from the humans earlier. Okay, so what are we looking for? Remember, we had actual human performance measured in D prime units, 0 to 6 here, predicted human performance of a hypothetical code. I've been talking about one code out of IT Cortex, but we tested many. So what we were looking for is if the code is correct, it should show that each task should lie on this unity line. That is, tasks that are easy for the human, say face versus non-face, high D prime should also be easy for the decoder. And if it's hard, then for the humans, it should be hard for the decoder. And it should, again, be on the unity line, meaning D prime of 5 is D prime of 5, so capable of actually meeting that level of performance. And so here we have a specific hypothesized code. So we tested many. This would be example, take IT data that I just showed you, but we also, again, tested other possible codes. To give you a sense of what we could find, some of which we did find, we could imagine this, what we call the correct code that I just showed you. We could have find something where all the tasks are poorly decoded. So even though the humans are good at some of them, none of them, a lot of them are sort of decoded at a low level. You could imagine that there's high correlation, but not good absolute performance, so that you'd have a good prediction of how difficult the tasks are, but you wouldn't reach the magnitude of performance. I call this an NIH happy code. And the reason you could have a pattern that looks more like this, where you have high performance on every task, regardless of what the humans do. So you're really good even when the humans are bad, and you're really good even when the humans are good. And I call that the DARPA happy code. Okay, so that's kind of a joke. I usually get a laugh out of that. But the reason I say that is because this sort of distinguishes between what maybe some people want to do with computer vision and what we are trying to do. This is what some neuroscientists try to do. We're trying to do this. So he needs to be performing and match the pattern, right? That's, to us, what success looks like. So that we want to be up there. Okay, so here's what we actually found. This is just one IT code. This is reading out of that 70 to 170 millisecond window with an SVM decoder. Here's the actual human performance. Each dot is a task. Here's the predicted performance from this particular code on all those different tasks. And you see these points like, it's not a perfect line, but they're very, very much along this diagonal here, are 0.91. Again, this is kind of what the ideal we might be looking for. We can't, we wouldn't expect a perfect correlation because, as I showed you earlier, even an individual subject doesn't perfectly correlate with the pooled average that we have here. So this is kind of the threshold. This is the best we could expect. And so I'll show you that quantified more in a moment. But you can see this is actually almost as good as that. And by comparison, you can look at, say, IT's input area, say visual area V4, and you see that it doesn't show this correlation. So it's not like every brain area is going to show this. Here's V4, an input area, read with a similar decoder style, and it has a correlation of 0.49. And I'd like to also point out it's not that it's just bad at everything. Sometimes it's too good at things, right? So it sort of figures out how to be too good reading out of these neurons in a way that the humans haven't figured out how to do. So that's what falsifies it. So if we now quantify this is just 2 example codes. Here's a bunch more. This is about another 10 or so codes, because we can test many of these ideas with different kinds of parameters. But I want to point out that the IT-based codes, there are some ways of reading out of IT with basic simple classifiers here. And I'll show you them here, that are actually in this human-to-human consistency band that I alluded to earlier, that the max we could expect is sort of in this gray band right here, given the variability in human-to-human consistency on these behavioral tasks. Here's some computer vision codes. These are some older things. SIFT, SLF is like a variant of HMax, a model we built, L3. Here's a V1-like simulation, like Gabor filters, a pretty good V1-like model, and here's some pixels. So you can see it's not like anything gets you up here. So it's by using these kind of comparison metrics that give us some confidence that this code actually might support that human behavior, at least the behavior as we've defined it so far. And you can then start to do even more precise things that start to say, well, what time window is that code available? What if we start to shift the time window around? And here's us shifting the time window around a little bit, and you can see that between the window of about 100 to 200 milliseconds after stimulus onset, that you're within this, anything read out of this time window is actually matched in its pattern of performance to human consistency. And that's very much what we might have expected, because reaction times in this task for monkeys and humans tend to be starting at around 300 milliseconds or so. So if you want to be supporting that, you better be available in this time window, and that's actually what you see here. Now, we hope with more precise tasks, we might even be able to get finer time resolution, but this is what we have right now. So many people are interested in things like the number of features. So how many neurons are you recording? Hey, you guys only recorded 300 neurons. How can it explain the behavior? There's millions of neurons in there. So I want to spend a few minutes to walk you through that a little bit. Okay, so let me take you through that. So what I'm showing you on this plot, this is consistency with humans. So that's that correlation that I've been showing on the last few slides. And I've added a measure of absolute performance now. So this is the average performance averaged over all tasks. So this is normalized against humans. So 1.0 means you're as good as humans are on average. That's the unity line on that scatter plot of tasks that I've been showing you. If you could get here, you'd be that perfect true code that we were seeking, what we're calling human-like. So just for comparison, here's some V4 code reads, and they sort of head up this way. As you add more neurons, you get higher performance, but you see their pattern never really looks like the pattern of humans, as I showed you earlier. These IT patterns, they start out at low numbers of samples. So this is with 16 randomly sampled IT sites, and you can resample out of our data to try many different variants of 16s to get an uncertainty on this that you see here. And you see it's actually not yet matched the pattern, and it's not nearly matched the performance. As you increase the number of IT sites, here's 64 sites, here's 128 sites. Pointer's getting a little, let's see if you guys can see that better. Sorry, that one's not working better either. You guys see that? Oh, that's a little better. So I'll just keep going with that one. So if you get to about 168, you're actually in the window where we can't yet, you could fool us as a human, right? You pass the Turing test because you're in the range of variability. But if you really want to hit the center of what we'd hope for here, that number turns out to be for us on the order of about 500. That's an extrapolation you can see from this curve here. So roughly about 500 neural features. We didn't record 500, but 168 is the max we had recorded here. So these are the visually driven units. So 168 or 500 uniformly selected IT neuronal features read out in that time window of 100 milliseconds using a simple decoder like a weighted linear sum like an SVM or even a correlation classifier will get you basically to human level ability and human pattern. Okay, so that now gives you a number here. As you can see, that number is driven not so much by this measure, but by this performance measure. So and in this audience, I think it's worth going through this number and performance in a little more detail than I usually go through with a pure neuroscience audience. And so let me try to take you through that here. So remember, we have to train the classifier. So typically what I've been showing you is training, in this case, on the order of about 100 images per object. So so when you have those binary discriminations, it's 100 per category. So we have 100 here, and then when I was telling you needed about 500, this is the number of features needed to reach that level of human performance, that reach that point, that pass the Turing test that I showed you in the last slide. Of course, if we use more training examples, we can get away with less numbers of neurons, and that's what this curve here is showing. If we use less training examples, then we have to start to extrapolate things even more, but this is the curve we estimate here. We think we're going to need more neurons to do that. So let me now talk about neurons. So these are trial average neurons, as I say, are multi-unit trial average. And so through a number of corrections that I don't have time to take you through, we can estimate what this amounts to in terms of real single units on single trial performance there. And that's what these numbers are here. There's an upscale here that actually brings you a factor of about 100. So that means that when I say 500, we're really talking about, to do this in real time, of about 50,000 single IT neurons. Sorry about the misspell. 50,000 single IT neurons to reach that point with 100 training examples to match everything as I showed you. And just to give you a sense of things, this is the max. This is the 10 million output neurons in IT cortex. Okay, so you see we really have to find a family of possible solutions here. I can't tell you this is exactly the way it works, but anything along here would work. We don't know how many training examples, say, a human would get to do these kind of tasks. Okay, so does this quantitatively predict behavior? I think I've taken you through a lot of evidence to suggest just that simple procedure that I showed you here suggests that this can quantitatively match things quite well. And again, back to all those hypotheses I laid out, I don't want to dismiss these as not interesting. They're all interesting in many different ways. I'd just like to say from a point of view of parsimony that these more complex alternatives don't yet seem to be needed to explain the behavior as we've measured it. So that doesn't mean throw them away, but that means make harder tasks, and then maybe some of these will become important. But I'll just sort of end by saying that's where we are with that right now. So why does the brain need to transform the pixel image? I've already told you that. Now I've told you kind of where the solution looks like it is. There is a solution in IT and the form that it takes. I basically told you that automatically evoked 100 millisecond time scale decodes out of IT with a lag of I was using about 70 milliseconds, are sufficient to support behavior. Sufficient to explain performance, predicts the pattern of human behavior in terms of difficulty. Okay, so from that, we think that IT population codes are, again, one simple step away from even human-level object recognition behavior. And although I didn't show you this, monkeys doing this task that we're doing right now in the lab show very similar patterns of performance on these same tasks as humans. So we have no expectation that something would be different in the monkey and the human, but we're now actually in the process of quantifying all that. So that was the first comparison that I wanted to take this group through. So I think I want to stop here. And people usually have lots of questions about this, especially non-neuroscientists about things. So I want to stop and pause and take some questions. Yeah. I'll try to repeat so folks can all hear.
Any idea why the V4 neurons are better than the IT neurons at least?
Yes. So it's the same reason that pixels can sometimes be better at some tasks. So, oh, I'm sorry. So the question was, why are the V4 neurons in some tasks better than the humans? And the answer is the intuition for many of us is like, try reading barcodes at the grocery store, right? We're not so good at that, but machines do it just fine, right? So those are tasks where we would call variation 0. So low variation. So the face is always front and center, and the face might face discrimination. The face will vary slightly, so there's some lowish level changes there that are still reflected in things like pixels and even V4 that can be picked up by a decoder, but apparently the humans are not as good at picking up on. So at least that's what's going on there. So the information is there, so when you decode it, they decode too well. They're sort of too good, and they've sort of revealed themselves, just like if I tested you to say, are you human or not, read this barcode, and if you were able to read it, I'd say, you're not human because I can't do that, right? So that's basically what's going on there. So this information, it's too good.
So just a follow-up to the question, is there any evidence, how much is that developed and how much is hard-coded? So if you change what the...
So you're asking, so the question is, how much of this is hard-coded, how much does this develop? So learning always is raised in these kind of studies here. I want to point out that, remember, we collect this data over many months. We don't see changes even within days or even over weeks of many of these neurons, at least just showing passively these images. There are some subtle changes that can be described, so I don't want to say nothing changes. But remember, these monkeys, we didn't have to train them to build up an IT. They come into the lab with a visual system, just like they come in with a V1 or a retina, right? So the neurons are already responding. Now, how that gets developed in early monkey life or through evolution, those are deep questions that we don't yet know the answer to. But the adult monkey, just sort of out of the box, if you will, through his natural visual experience, already has a representation that is automatically evoked by even like images of cars. So just because the monkey's never seen a car before doesn't mean it's not has visual statistics that aren't going to be represented in this basis. So I don't know if I took your question as a chance to deviate in another topic, but did I answer it or should I try a different tack? Okay. Dan? Yeah, so we would love to. So you can start. Some people are now doing like 2-photon imaging in parts of early monkey cortex. That it's very hard to do in IT right now because it's very deep down. So I didn't show you in the surgery. You're basically down by the ear. So we would love to do that. You're only going to be able to do the surface, but with those arrays, you can only do the surface too. We haven't done it yet. I mean, it's also intriguing that so far with these methods, we're actually already still matching things, but we would love to do that. You could do fMRI, but that's just worse. So some form of imaging, especially things like 2-photon, would be the potential next generation, but we're not there yet. Yeah. But it would certainly be awesome. And, well, maybe we should talk after. Yeah, okay. I see lots of questions, so I don't know, go. Yeah. Oh.
Fascinating. Sorry, someone else?
Nico, yeah.
Fascinating aspect of this is sort of the search through the space of possible codes and where you find sort of the one that best predicts actual performance. Could you talk a little bit about that search space and how do you cross-validate then in order to estimate this?
Yeah, I think that's right. So I think that's right. So it's like the more codes we search, then we have to start having a new validation set. So really, these were the codes that we kind of planned to test out of the box. So that's why I present it this way. Now, in practice, we search the numbers of neurons, right? But it doesn't matter which neuron. You're not overfitting there, right? That's sort of, I think, well regularized. So that's one of the main things: the number of neurons, time window. But I mean, I could show you a plot of every code that we've tested, but I don't think.
Is it thousands or is it?
It's I don't know if Ha's in the audience here, but it was sort of on the orders of at least hundreds we've tried. But I didn't try hundreds and then pull one out and say, aha, right? So I mean, I'd already showed you that study I did with Tommy was basically the same code, right? So that was the main one we wanted to try. But then of course the guys went crazy and said, let's sort of search the space a bit. And it turns out, nothing dramatic fell out of that. It was just the rate codes counted over that time window is roughly in the ballpark. And, we were exploring the number of training examples, and that mostly just moves the performance. The pattern in IT matching humans is very robust to a lot of these decisions. It's just when you drop the number of neurons or make the time windows too narrow that you fall off the line, as I showed you, that you should have dropped the pattern at that point. But most of the ways we're kind of, well, let me just stop there for now. Maybe you and I can talk afterwards. Yes?
So in the plot that you show D prime of the classifier versus human D prime?
Yes. I could try to back up here a bit. D prime of the classifier versus human D prime. The scatter plot, yeah. Is that what you mean?
Yeah. So in that one, you average over all task difficulties per task, right? Can you do some final analysis there and say, for example, you see if the task gets more difficult than humans and that would become...
Right. I think maybe I think you're saying, did you average over...
Yeah, exactly.
We averaged over images in effect, right? So I think that's an important point. So each of those dots was a task. It was like car versus no car. And there were many images of cars, right? And I just give you one number out of it. I don't tell you the performance on each image of the car. I just say, hey, car versus no car, here's how hard...
Just changing the level of the task difficulty. Like variation.
Oh, that was actually in there, and I kind of showed that in the human data. So those different tasks, this was car versus no car at high variation. Then we had car versus no car at low variation, and that's partly what gave rise to that large scatter. And maybe back to this earlier question about what is V4 doing, those are those low variation tasks that V4 sort of discovers, oh, I can do really well at this, but the human doesn't see that. I thought you were going to point out something important that we haven't done yet that we want to do next is we start looking at image by image predictions. We don't want to declare victory here and just say, oh, let's all go home. Now it's like, can we dig deeper by looking at the patterns of errors on individual images, which is a much next level of analysis. So first there's average performance, then there's pattern of difficulty across tasks, then there's sort of pattern difficulty within images, and so that's what we need to do next. But the more you do that, the more data you need from neurons and monkeys, in humans. So.
One very quick question. So why did you go after binary tasks and why never say 4 classes and then look at the confusion matrices and then do some analysis?
Well, okay, so we did do an 8-way task. So we had an 8-way confusion matrix. We just thought it was easier to do the analysis to then from that recover the binaries. But I don't think it matters much, but I think maybe we should talk about that offline too. But we did actually get the 8-way confusion matrix. Yeah, I think I see this hand up and maybe.
How do you think might the animal's measured responses depend on the animal's experiences, its memory of its life?
So that was related to how do the responses in the neurons in IT depend on the animal's life? And that was related to the learning question I had earlier. There are learning effects in IT, but again, what I've shown you here is an adult animal that wasn't trained to do anything except fixate. So this doesn't mean that we couldn't bend these neurons. In fact, we know and others know that you can, under certain training conditions, bend these neurons. So I don't think it's sort of a totally different orthogonal question of how learning affects these neurons. What I'm hypothesizing here is the learning going on here is downstream of these neurons. That's the hypothesized decoder. Like prefrontal cortex, other labs have suggested that's where these category units may be formed. And what we're saying is there's a basis, a shape basis, that exists even in an adult animal through whatever visual life experience it got, and that's what's being decoded. Whether that, if the animal had been raised in a non-natural statistics environment, we would expect that basis to be different. That's a really interesting study, but nobody's done that experiment yet. We don't know the answer.
You can train the animal, but the animal is already trained in its environment.
Yes. So the animal's seen a bunch of natural statistics, been under whatever goals the animal's had for its life, and that's what we get. And we haven't tried to control that at all. And that's where we are. So certainly you could do those things, but we didn't do that in these studies. We've done things like that in other studies. And again, maybe we could talk about that offline. I think maybe I could take one more, and then I should maybe move on, or we're going to run out of time. Yeah. Wow. Okay. Yeah. Go ahead.
Have you looked at this performance comparison in any areas immediately downstream of IT?
Yeah, that would
be interesting.
Yeah, I think that's a great question, and that's sort of like searching for. We're kind of hypothesizing a unit downstream, but we haven't looked in. Like prefrontal would be an obvious area to use. Look, or perirhinal. We haven't yet. So, I mean, I think from an engineering point of view, we're kind of saying, well, this is fascinating that we've already got a good feature set, and we know how to build linear decoders. So it's like the feature part of the problem is where to us is the interesting computational part. But that's a really good neuroscience question. That
one might expect that actually performance could fall, right? As if there's specialization.
Yeah, one could also expect that we're going to run into the training issue that was brought up sort of in these other questions, that the monkeys were not trained to do any of these tasks, right? So again, nobody, if I was up here talking about reading out of the retina, nobody would say, oh, you better train the monkey to build up his retina. They would just assume the retina is sort of already somehow built correctly, right? And so I'm kind of saying the same thing in IT. It's like it's already built correctly for somebody else to look at it, right? Okay, so I should probably move on, and then I'm sorry, and the questions like that, I can take them again on the end if we have time, but I want to get through the next two parts of this talk. So I think I'm an hour in at this point. Okay, so let's shift gears, and we're now going to do comparison of machines and monkey neurons. So this is now an attempt. What I'm going to take you through here is to try to show that a focus on modeling the behavioral goal, that is invariant object recognition, is leading to what we think is a new way of understanding the underlying brain mechanisms. And I'll show you what I mean by that, I hope, as I go through this. So the question really here is a sort of good segue from the last question. We think simple decodes from here are sufficient to do a lot of the things that humans do. But what we're interested in is what's the transformation from, especially from area V1, where we think we know a lot about what these neurons are doing, to up to IT. So what is the transformation that goes on here? And to us, this is where neuroscience meets computer vision, where people are also struggling with this problem. What features should they build? And we look to that field for inspiration about what kind of features to build. And so that field, for a long time, it's been driven by neuroscience-oriented models. And we started exploring some of these ourselves. And this is our work roughly in 2008, to introduce you to a basic model class that we use that I think is very similar to a class others use. And it's basically a set of dot products, so filters. So you could imagine Gabor filters for V1, followed by threshold nonlinearities, like a rectified nonlinearity, perhaps with a ceiling. Some pooling operation here. You could think of max pooling or other types of pooling. Then you had to have a normalization operation. So these are things, the operations that many people use in their models, both neuroscientists and computer vision folks. This is just a one-layer model here. So you might have different levels. Here would be different oriented filters in this example. And so you might have thousands of output features in this kind of model. We, in our lab, have been especially interested in these so-called architectural parameters. So the filter parameters are often a deep point of interest for folks interested in learning. There's another part of what we call learning that has to do with how did you decide to set these thresholds, these poolings, the strides of these things, the normalization strengths, and all the other parameters that we lump together here as I'm calling architectural parameters. So these thetas, there's not just 4 of them. There's a large number of parameters in here. This is just to give you a feel for what's going on. And so what we did first was we optimized some of these architectural parameters for performance on a one-layer model that we call a V1-like model. So we had Gabor's up here, and we optimized some of this for something called V1-like model. And I'm just introducing you this to give you a sense of kind of how we got where we are right now, which is this is Dave Cox and Nicholas Pinto when they were grad students in the lab. And this is a while back on Caltech 101. Nobody really bothers with this benchmark anymore. But in 2008, this was kind of people were competing on it. And what we found, to our surprise, was this V1-like model that we considered to be a neuroscientist dumb null model. It's just a one-layer thing. Was already kind of outperforming computer vision models at the time on this kind of benchmark. Now, we didn't think that was because we had this great model or we were great at computer vision. We thought that was because the benchmarks were just really not very good. That you could say, well, look, car, there's no variation in here. Cars from the sides, correlated background, there's lots of ways to cheat. And so this kind of set us off on this idea. We need to have better benchmarks if we're going to actually make progress in this field. And so we went off, while many folks have gone off and done millions of images, and that seems to be where computer vision has gone, we went off in a different direction, which is to say, look, you want to be able to do this kind of task, We are going to do this in a more controlled manner. And we're not the first to do this. I know this is kind of out of favor in a lot of computer vision. But what we did was we took 3D objects. We took the view parameters I just said earlier. We rendered them. We placed them on naturalistic backgrounds. This is just a review of what I showed you earlier on. And we did this because we'd have parametric control. Somebody mentioned in the question we could go from low variation to high variation and make tasks gradually more difficult. That's nice from driving model performance. And, okay, I already showed you examples of this, car versus not car. I should have shown you that. We could do few images, and we could easily see that the computer vision systems at the time, we could easily crush them with this kind of task. So even though they were saying, look, we're doing great on Caltech 101, we could sort of easily push them down with this. So that told us, look, you don't need millions of images to push them down. Here's an example of that V1-like model that we said, well, look, we're state-of-the-art in Caltech 101. As soon as you increase the level of variation going up this way, you quickly drop in average performance, and here's humans on those same kind of tasks. So this kind of points out a couple things. First of all, if you test in these low variation regimes, machines are actually already better than humans. This is like barcode reading to us. This is okay. You don't want to claim that you've successfully beat humans out in this regime. Here's where the action is. This is where machines lose to humans, and this is where there's something interesting to learn from the brain. And these kind of stimulus sets could easily expose that. So we still use these stimulus sets. You heard about them this morning, and because we collected a lot of neural data on them, as I just showed you. And so this stimulus set, we call this set HVM 1.0, which stands for Human versus Machines 1.0. And this was meant to test what we're calling Object Recognition 1.0. And here's now the performance. Here's the same stimulus set I showed you earlier. And what I want to just point out here is this is a plot of different models and their performance on the set at high variation. And this is just making the same point that they're all very, very poor. These are older models now. These are like HMAX, our V1-like model. Again, this is 2009, so there's a bit of history here. Here are those IT units I just showed you, not extrapolated. This is 168 neurons, and here's humans here. So again, you can see everybody's doing quite poor relative to humans. Again, we didn't need a million images to actually show that these models are not as good as humans. We could just use that kind of set. Okay, so it was clear, even on that kind of simple-ish, smallish set, that these models are not doing what humans are able to do easily. And so that then kind of gives you two choices if you want to improve things. So possibility one is, say, the model class that we and others were starting to work with, this feed-forward, largely feed-forward class, HMAX being an example of that, was somehow just deeply flawed, and we had to start putting in feedback and do something more interesting there. Another possibility is, well, there's a lot of parameters in these models, and we just don't know how to find the right parameters. And so we, again, I think we're kind of a parsimonious group, and we said, well, look, let's kind of work hard on B before introducing this, because if we do this, it's going to have even more parameters. So let's try this a bit harder before we sort of move to the more complicated models. Not because we don't think there's any feedback in the brain or we don't think it's somehow important. We just didn't think this had been given a sort of fair exploration. So what I'm going to show you next is our overarching strategy for the last couple of years is to start with this largely feed-forward, bio-inspired model class that I introduced you to briefly with one level. And then we want to try to optimize performance on the tasks the brain's good at, i.e. the ones I just kind of showed you, which easily separate brains from machines. And then ask, do the model features look like the brain features? So that's what we're going to do here in this set of comparisons that I'm going to make next. Okay, so now remember, it's not just a one-layer model, so now we can call this a deep model. Jan says as long as there's 3 levels, I can call it deep. So there's there's at least that's what he told me. Okay, so it's got 3 levels, and this is a model class that we were using in 2009. This is Nicholas Pinto and Dave Cox doing this work at the time. And I want to point out it's deep in that sense. It's convolutional. Again, neuroscience inspired those convolutional models, and they were LNN units. That means the linear operations with nonlinears here. Neuroscientists tend to use those units already. And limited feedback. And I say limited because when you do normalization in a model, that has some feedback to actually execute that operation. So this is a class of models. Again, it's inspired by many others, folks like Kubel and Wiesel, Fukushima, Tommy Poggio, Jan LeCun. Many have used this kind of class of models. We extended it and parameterized it and implemented it on things like GPUs so we could explore the architectural class. So we were, again, especially interested in these architectural parameters of these models. And I remember the filter parameters are often of interest to a lot of folks, but we were focused on these first. We thought these are the sort of big things we need to try to sort out in a way. And the reason we got excited about those parameters is because even when we set the filters at random draws of the random, so initializations of the filters randomly, and we just played with the architectural parameters, that we could get pretty large gains in performance just by optimizing our choices of architectural parameters. I showed you that with that V1-like model. Nicola and Dave did that later with these deeper models, and they were obtaining state-of-the-art or above state-of-the-art performance on things like phase tasks, and they went on to do a lot of work in that line using this kind of strategy. So, but the point I want to make to you guys is that if we just look at the architectural parameters, there's stuff interesting going on there. Even without optimizing here, learning about these we thought was already an important thing to do. And this is then work from Dan Yeamans, who's here in the conference, and Ha Hung. What they noticed was that with different choices of architectural parameters, some models, so you set a parameter, random filters, choose your architectural parameters. Some models, some choices of parameters do better on some tasks. For instance, here's an example, faces versus tables. There's a model. This is an exploration of models. Each dot is a different set of model parameters. Here's performance on this subset, this task. Here's performance on this task, fruits versus tables. And you can see, well, there's a model over here that does well on that, but not so well on this. There's a model here that does well on that, but not so well on that. So that suggested that, well, we could maybe make mixture models where we took different choices of parameters and assemble them in a mixture way where we're going to put them side by side here, can organize them and say, well, look, this is all still part of level 1. We're just going to choose them so that they're dealing with different parts of the object space, in effect. And so this is what Dan Yeamans really led, and he did this, partly he was able to do this with an optimization procedure that was developed by James Bergström working in David Cox's lab called HyperOpt. And I'm not going to take you through the details, but Dan has a poster if you want to hear more about that. But basically the idea is you have a performance goal. I'll show you what our performance goal was in a moment. And you have a set of architectural parameters here, and you start somewhere, and you're moving around in various ways to try to optimize that. And along the way, you're going to draw off different models that do well in different parts of the task. And this is basically ADA boost at that point. And then after you collect a few of those, you're going to go and do this. You're going to do a reweighting of the performance. You're going to re-optimize again now on the tasks that you're still, the portions of the problem that you're still lacking on. This is on a screening set that I'll show you in a minute. And you're going to collect some more components here. And then you're going to assemble these kind of components. And then you're going to repeat this. So you're now going to sort of take the outputs of this as a new feature set. And then you're going to repeat this whole procedure again. And so Dan so far has just repeated this once. So there's just another level on top of this, as you sort of see here. So this is just the model that he built so far with that idea. Dan calls this hierarchical modularization, HMO. I want to point out that we're not really wedded to this optimization procedure. We thought this is just one way to explore the architectural class. And so here are the screening sets that Dan used. These are the model screening images and tasks. He had basically 36 objects. These are three-dimensional objects that, again, were rendered, placed on naturalistic backgrounds, control the amount of variation using the same strategy we did before, but now these objects are completely separate from the ones that I'm going to show you next, the ones I've already shown you that we're going to test on. So we optimized a model on these kind of things here. And he did that once. He went through an optimization run and got this first model out that we call HMO 1.0. Let me show you HMO 1.0. This is the performance on this kind of task. Remember, I showed you could bring computer vision systems at the time to their knees, at least the ones we were testing at that time. And again, here's low computer vision systems. Here's human performance. And then HMO was able to match human performance, or very close human performance, even on these high variation tasks. So that said, just by using the screening procedure and then going over and testing on these kind of images here, at least in this kind of domain, this model was able to do quite well. So we'd met our optimization goal. Let's find higher performance. Now let me sort of say, okay, now that we had a high performing model, remember our strategy was find a model that performs well and then ask, does it look like the brain? So we optimize a model. Here's the model, 4-layer model with the components I mentioned earlier. We optimized it for a set of screening tasks. And then we ask, does it look like the brain? So the first obvious thing we did was we took the top layer of the model and say, does it look like IT? Now, how do we ask if model features look like neurons? Well, we have to take one single neuron here. We're not claiming, we wouldn't know what feature in here corresponds to that neuron. But what we'd like is that this feature set, we can make a linear projection from here to here, that these responses live in the basis defined by these features here. So we're just going to do linear regression to fit each neuron. And so we use some of the response data collected from each neuron as the training data for the regression, the fitting data, and then we use the rest of the data as the test data. And I'm going to show you the predictions on the test data. So again, this is cross-validated. Here's an example. So this is an IT unit. So this is an IT response. The black is the response of the unit. The images are shown here. So you're getting one value out of the neuron for each image. So remember, we're just counting spikes in that 100 millisecond window that I showed you earlier. So you see that they're grouped here by semantic category. You can see this neuron is semi-semantic in the sense that it kind of likes some of the chairs more than other things, but you see things are bouncing up and down. This is typical of IT neurons. They're not purely semantic, but they have some hints of semantic structure here. You might say semantic or shape. We can't say which. They're highly confounded here. But you have this, what I want to point out is that you have HMO shown in red is very overlapping the black here. The correlation, the R squared, is 0.48. So half of the explainable variance is explained by this model. And that's a huge jump relative to other models that were even trying to explain earlier visual areas at the time, like input to IT, only like 20% of variance explained on, for instance, area V4. So that was impressive to us that all we'd done was optimize a model to do well in the kind of tasks I showed you, and then took the top of the model and made a linear projection onto IT neurons, and suddenly we're able to fit these neurons much better than any other model had ever been able to. Here's another neuron, 0.55, really good. This neuron tends to like faces, as you can see here, and as I mentioned, there are known sort of face neurons, or so-called, quote, face neurons in IT cortex. Here's another neuron that's not clearly semantic, just to give you some sense of it. And you can see this still fitting quite well if you follow the red and black or over on top of each other. Here's an example of another neuron, just to give you a sense of, we tested other models. So here's a V2-like model, and you can see it sort of underpredicts here, and then it makes some funny mistake here on this image. Exactly why, we didn't track that down, but just to give you a feel that HMO is doing better than these models. Of course, we quantified all that. Here's HMO's top layer. This is a distributional model, fits over all the neurons that we had. So each bin in this histogram is a single neuron. I showed you a few examples. We quantify things by taking just the median here, which I'm showing on this plot. So the median explained fraction of variance is about 50%, 0.5 here. That's what I mentioned earlier. So 50% of single response variance is explained on average. And that's a dramatic improvement over all previous models. Here you can see a bunch of other models that we also tested. These models, of course, were given the same procedure. HMAX features were given a chance to linearly regress using the same data on the same neurons. You can see their distributions out here. Same for all these other models here. I want to point out, so the HMO model, you can see you actually looked at the lower levels of it. It's just the top level that does the best so far relative to these other levels, which is what you might expect. I want to also point out that Dan and others did a nice control where they did something where they took just the semantic, the labels of the classes and said, how well can I just fit IT neurons by using the labels of it's a car, it's a chair. So of course that's not an image-driven model, but it's a model to compare with. And you can see that that explains much less of the IT neurons' response variance. What that means to us is that the model class that's been built, or that we used, plus the optimization criteria, that is the kind of tasks we were using, are somehow inducing structure, that is the features that come out of them, beyond that are induced by the task. So it's not just that we're producing this. There's something going on in both the brain and the model that are matched so that our performance is well up here and not down here. Okay, so but the big take-home message is that we are getting more dramatic improvements in our ability to fit IT neurons by just optimizing for performance in this kind of model class. Okay, so that was just showing you single cell fits. But we, as thinking about population decodes, we have a natural tendency to not want to say, oh, explaining single units is the goal of what our lab is trying to do. As I showed you earlier, population decodes are what give rise to the matching of performance. And you need hundreds of sites to start to match that performance. So we're especially interested in measures where you can compare one population of neurons to another. And we really like this technique that this is introduced by Nico Crisgorte, who now I noticed is here. And Nico introduced this basic idea that you have the responses of different neurons. So this is like the state space I showed you earlier, response of IT neuron 1, say, neuron 2, neuron 3. And what you're going to do is think of each image as living in this population space, just as I showed you earlier on. And you can imagine thinking about, hey, these images are closer together than these. And it's sort of the relationship among the images in the population space that you would hope that if you had another basis that it would maintain those relationships. So it could be a rotation of the space, for instance, or some scaling. But as long as it maintained the relationships, you would say this is sort of equivalent to that. So what we want are measures that can kind of just capture this and be essentially tolerant to things like rotations. And so this allows you to compare one population to another, as I'll show you in a minute. So if you look at the layout of images in one space versus another, then you can begin to ask, are they similar? And Nico did this, they call the technique representation dissimilarity matrices, or RDMs. And here's an example from their paper. What I want to show you here, don't worry about the math. I just want to show you that you basically look, blue means that two images are close together. So You think of each of these pixels here as like a pair of images. So that image and that image. Blue means they're close together, and red means that image and that image are far apart. Okay? And near and far here are measured by correlations, or dot products. And so this is just the layout this is, I think, I can't remember if this is IT. I'll show you in the next slide if that's IT or human LOC. It doesn't matter, as you'll see, but that's a population representation from Nico's paper. So RDMs, the reason they're important is that they allow the comparison of any two feature representations on a common stimulus set. So now we're looking at things at this population level as a whole rather than just fitting single units. So this allows you to say, hey, how does monkey 1's IT look like monkey 2's IT? That's kind of a key question. How much individual variability are there? You know, I can't assign a neuron in monkey 1 to a neuron in monkey 2, but I can ask how similar their population representations are. Or how much is IT actually different from V4? Or how does IT in a monkey look like versus a model, which I'll show you in a moment? Or how does monkey IT, and this is what Nico did, look like relative to human IT? So this is from their paper again, and you can see that these actually look quite similar. These are human fMRI voxel activity, and this is monkey IT. And they point out that these are actually quite similar, and that sort of is partly how we know there's a good homology between what's going on in the monkey and what's going on in the human. Okay, so now back to our data using this technique. So these are RDMs now for monkey IT neurons. And so these are the IT neurons that I showed you earlier, now on these images that I showed you earlier also. So what I want to point out is that you see this block diagonal structure. These blue blocks mean that the neurons somehow have arranged the images in a way that it makes it pretty easy to decode the category. And we already know that because I showed you earlier that you do pretty well on these tasks decoding out of IT neurons. But now you can also see there's interesting structure in the off diagonals here about what's near what. See blue and red over here. There's some structure over here that now we can sort of ask how well does that structure repeat in, say, for instance, a model. And just for comparison, here's V4 on the same exact image as you see less of the block diagonal structure. And that goes along with our knowledge that V4 is much less good at doing many of these high variation tasks than IT. Okay, so all this was a long way to just introduce you to the idea that there's a population way of comparing things that might be a more appropriate level of abstraction than comparing single units. And here's what happens with that HMO model that I showed you, that Dan Yeamans built that I showed you a little while back. So here's the HMO model. Here's the monkey IT using the RDM technique. What you should be looking at here is like how similar does this look to this? You can look at it with your eye. You can say, well, what structures are that is over here. I see this blue and blue over here. So you're seeing the similarity of structure. We can quantify that just using a Spearman correlation here. So this is the Spearman correlation that quantifies the similarity of this matrix to that matrix. So just compare those directly. Here's HMO versus that. So that's the number right there. You see HMO is very, very high, much higher versus IT than any of these other kind of models, things like HMAX, a V2-like model, a V1-like model. So they're all way down here and actually almost near the IT versus IT ceiling that is the limits of our data that we can't expect it to really exceed given the current volume, the amounts of data that we have. So this is just another, maybe a long-winded way of saying when you look at things at the population level, this model looks even more like IT than when you look at it at the single unit level. But it's just another way of comparing models to neural populations. If you like this, it's nice to sort of lay out what we think is going on at these different matrices, different levels of the ventral stream. Here's IT and V4 I already showed you. And you can see you get down to pixels and you lose all the block diagonal structure that you see up here that you get out of the HMO model. Okay, so I've compared the model with IT neurons and said this model is explaining a lot of the variance at IT at the single unit and the population level. But we can also go, well, if this model is actually sort of telling us something correctly, well, what about its intermediate levels relative to the intermediate levels of the brain? Remember, we collected V4 data as well. So now we can take the intermediate levels of the model and compare with V4. So that's what Dan did. Here's the IT fits. Now this is back to the single unit data that I showed you earlier. So here's that explained fraction of variance. Again, I already showed you this before. Here it is now against V4. So this is using the exact same techniques. You take a V4 neuron, you build a regression onto it, you cross-validate with new images. How much variance of the response of that V4 neuron do you explain? That's plotted right here. And what you can see is that this model at its top level explains only 30% of the variance, but at its intermediate level is explaining, again, near 50% of the variance. So level 3 of that model happens to explain near 50% of the variance, and then it drops down a little bit down in level 2, but all way down in L1. And again, this L3 is much higher than a bunch of these other models, and also much, much higher than this category-like model that I talked, not image-based model, but category regression that I talked about earlier. So again, what we think is going on is that the model class somehow, remember, we didn't build the model to say optimize to build V4 neurons or IT neurons. The model was just trying to do recognition on the kind of tasks that I showed you in a structure that we thought was bio-inspired. And it turns out that even its intermediate layers match quite well to the intermediate levels of the brain. And in fact, not only quite well, but they get much dramatic advances over previous models in both V4 and IT. And so we think we've kind of found a new strategy to kind of combine model classes with an optimization approach for tasks to get us to actually explaining neural data rather than directly fitting the neural data. Okay, and let me show you that in a quantitative way. So here's the performance of model features, and here's the ability to predict, this case is IT single unit responses. So remember, a fraction of explained variance. So what you see here is that these blue dots are samples of the bio-inspired model class during an optimization run. Each dot is a different set of architectural parameters, so a different model, if you will. And again, it's tested for performance and its ability to fit IT. And what you see is this correlation. And you also see that even in the existing models like Pixels, HMAX, some of those other models that I've been showing you, there's this rough correlation. So what we've been doing is optimizing performance of our models. That's what I took you through, optimizing to build that model that we called HMO. We were just trying to optimize performance. And in 2012, then we had that HMO model, and now it's explaining 50% of the variance. So we went this way, and that lets us go this way. It turns out, Dan's also shown that if you just try to optimize directly this way, you don't get any better than going this way. So this way gives you both performance and fits. And so we think a strategy going forward is to just keep optimizing performance, and that we're likely to get even further improvements in fits, hopefully of IT and V4. So I want to say, I know I went through that quickly. Dan and Ha are both be at their poster tonight. If you're interested in this kind of modeling effort and its relationship to neurons, I encourage you to go by their poster. Any of the other things I'll tell you, they can talk about that too. T63 tonight. So please put in a plug for them if you guys are interested in that. And they'll be able to give you all the gory details. Okay, let me stop right there as I still a half hour or so left, right? So I think this is another good time to stop and take some questions on that part of the talk. Or on the last part of the talk. Yeah, I see one in the back, yeah. So remember, the results, the question was, how sensitive are our results to the number of electrodes we use? So the results I just presented here, we're not doing anything to optimize neurons until that last regression step. So I think your question might be, would our 50% variance number go up or down if we had collected different neurons or more neurons? Is that, I mean, we don't really know that, of course. I mean, I showed you the distribution of the sample. There could be some pocket of neurons that we didn't record, but it's sort of guessing about a distribution that we just don't know. We don't suspect it would change that 50% number, but other than looking at the data sample that we have, I can't say more than that. I wouldn't expect it to change dramatically.
Are all the electrode tips at the same depth?
They're built to the same depth, but the surface of the cortex is a little curved, so when we inject those things in, they're probably at slightly different depths. And I want to point out, actually, these are multi-unit data often that we're fitting, and I probably should have been more clear about that. But when we sort single units, we get very similar results. Because remember, those arrays, they're in for a long period of time. They don't always isolate good single units. Other questions on this? Yes?
Simple question. So this, the code that is in IT is already a code, right?
Well, I call it a code.
Yeah, I mean, so you decode it, your algorithm. So where is this decoding happens with motor neurons? Like the animal should press something, right?
Right. So that's I kind of punted a little bit on that. The question is, how does the IT get out to the motor system, right? So I sort of said, look, we can put a classifier on it and then match the pattern of performance. And that's essentially a hypothesis that somebody downstream is going to do that kind of operation like we're doing. And it's an operation that downstream neurons could perform. It's a dot product. But is that done in prefrontal? Is it done in the motor system? This was sort of related to a question I got earlier. We didn't investigate that. We're just saying the basis is sufficient that's a viable downstream hypothesis. That's the extent of what our data can say.
Is it possible to check it with current neuroscience techniques? Like this decoding happens exactly.
Well, yeah, so the closest, and the best work I know on that is Earl Miller's work on categorization in prefrontal cortex. You train a monkey for a long period of time to do cats versus dogs, and then you can find neurons in prefrontal cortex that can discriminate cats versus dogs. And yet IT cortex maintains some more of a shape basis. So our work is synergistic with the idea that, well, if I train the monkey to build do the car versus no car, he would build car neurons in prefrontal cortex as linear decoders off of the IT cortex. And what we're showing is that this works even in these challenging tasks, and it matches things quite well in terms of the predicted patterns. Does that okay. Any questions about the modeling part that I just went through?
So it seems, you are saying from these matrices that you get the invariance, the object invariance, you get it sort of for free?
Yes, right. So the IT already has the ability to say be trained on a small set of images and then generalize over other things that we're calling invariance for free. And the matrices imply that, but I think the better test is just doing the straight decoding, and that's what we did in the earlier parts of the talk that I showed you. So yes, this basis is a basis that you need few training examples to be able to do those high variation tasks.
But basically you get very close responses for small chair, big chair, rotated chair.
They don't have to be close. They just have to be such that, now we're getting into like what your training sample looks like. But so you give me a few examples of like a car to the left and the right, and then I'm going to know I need very few training examples to put that hyperplane in such a way that all the cars are going to be correctly classified. So that's the idea of the identity manifold being more flat and enough that you can find it with low numbers of samples. So you don't need to use large numbers of training samples in an over-complete space to find it. So yes, you get a lot of invariance not for free, but almost for free. But you need some training to get it. And now we can get into details of how much training under what conditions. And I didn't try to take you through that. But yes, the short answer is you get invariance for free out of IT. The short answer is yes.
It's all ipsilateral, monocular ipsilateral analysis.
Well, okay, so there's some subtleties there. So he's asking, is it all ipsilateral? So many of you know, so we actually recorded out of both hemispheres of IT and V4. And then we worry more about this in V4 because IT fields include most of the center of gaze. But when we want to make comparisons with V4, we do things like only consider when the objects are in that field or only at the center position to make sure we're not falsely concluding something because of the failure to cover the retinotopic maps fully.
I just wonder if the volume image, the stereoscopic information improves
Well,
if you went simultaneously in both hemispheres.
So you're asking about three-dimensional, like depth information now?
Rotating a chair, a flat chair in an image is not a three-dimensional volume chair. The shadows, the depth, the converging lines.
Oh, you're asking about our rendering now? I mean,
Just the value of more information.
Okay, so maybe again, when we do the training here out of IT, we're training with static images. So that partly what you're saying is like, well, what if you had temporal images and you trained off of that? And we have some other work on that and how IT neurons sort of may change in interesting ways there. But we didn't do that here. So it's true that, yes, when you're learning, having time to sort of see the sequence of chairs might help you in ways that we're not testing right here. Yeah, one. I'll take you through those other models. That's the next part of my talk a bit. But the short answer is that we recently have determined that they're in the game with this model. So about half the variance explained by the Krzyzewski model and Rob's model with Zieler. And I'll show you the performance of those models relative benchmarks next. If you were at Rob's talk earlier, you've already seen what I'll show you next. But the short answer is yes, 50% of the explained variance from those models so far too, which supports our kind of overarching hypothesis that if you optimize these models, I mean, they didn't know anything about our neurons. Even Dan maybe knew about our neurons, but they don't know anything about our neurons. So the idea is if you're optimizing models to do well on these tasks, then they kind of naturally start to fit these neurons. Maybe that's not surprising. It's saying we've sort of found the same joints that evolution's been working on or the animal's been working on or some combination of both under an architectural constraint that we've gotten close to. That's the way we think about it. And at some point, that might break, but so far it's working. Yes. Yes.
Have you attempted to classify objects that are different from your training set using your already trained model? So in other words, are objects
So you're asking about the HMO model now and its ability to
You have chairs and bases and things. Is there some geometric distance you can construct with nothing to do with the classic models, and then use to interpret the model activity to classify these objects without showing them as training sets?
That seems to Dan, Dan's here, I think. So that sounds like a question you might be better suited to answer.
Can you generalize to new classes of objects without training them? In other words, by exploiting the similarity between objects, can you generalize to classify new objects that were not shown to the monkey, that were not trained against?
You mean if you,
Like
if you pick like, I don't know, some number of object centers, decide that is your universe, and then you show me a new image, and just work on some kind of like distance and feature space, is it going to classify that image correctly?
Yeah.
Well, I mean, it will if the object is in one of the categories, presumably that you've used to like pick the centers. I mean, I don't fully understand. You're wondering
I'm
whether or
wondering
not.
if you can generalize beyond just the set of things that you train against. And if so, can you do that by looking at a distance measure in object space? So some new class of objects have the distance from that class and existing classes you trained. Therefore,
But if it's new, right, then like presumably if you're expecting it to give an answer on something among labels that you've already chosen, and you know you're telling me by definition it's a new class, then obviously it can't report that new label. So obviously not.
no,
almost
you
like
could interpret your model if the activity were somehow transferable.
You may be asking something like, if you do give it a new class, so like chair prime, that's not exactly chairs in some way, but it's similar. Will it be the case that like the distances in that space and the feature space predict human confusion? So humans might be confused between chair and chair prime a little bit more than they would be between, say, chair and chair prime on some other set of things. And your question is something like,
Maybe we should take it offline.
Yeah. I mean, this might be partly answered. I held this slide back. This might be related to your question. So here's the RDMs I showed you of HMO versus neurons. This is with image generalization. So this is meaning you took the HMO representation, you trained to fit the IT neurons using some images, and then you took the other images and built the RDM out of that. And that's what you're seeing here. But you could also do things like object generalization. Let's say category generalization. That's the strongest, right? So you're just doing the projections. You're just fitting the IT neurons using the basis set from HMO based on not these 4 objects, but the other 4 objects. So I don't know. Here's 4 objects. You got to find the other 4 here. So you just do those 4, and now I'm showing you the predictions for these 4 objects. So the layout of these 4 objects in this RDM space of the HM model and of IT, and you can see that there's a lot of structure here. And in fact, if you quantify that, it's actually still very, very good. That's this line here, and much better than other models. So this is one form of the generalization I think you're asking for here. But maybe you guys can take that offline if that doesn't answer it, because I'm not sure I understand the question fully either. Let me just, since I'm now, I think I've only got about 20 minutes left or so. Let me take you through, this won't take very long, so we'll have time for the questions at the end. And Rob went through this earlier if you were at his tutorial. This is now comparing machines versus monkey neurons and human behavior. And really the upshot of this is it demonstrates, I've been talking about a lot of computer vision models not being up there, but of course I've been ignoring the recent models, these recent bio-inspired models, that they're now rivaling the brain in object recognition. So, and I'm going to show you that now. So this is the work of Charles Cadeau as a postdoc in the lab. And so what about these other high-performing networks many of you heard about recently, deep neural networks? And I'll show you Charles's work here. Again, I'm referring to the Krzyzewski work, so I thank Alex and Jeff they ran their model on our images that we sent them, and Zieler and Rob here for doing the same with their 2013 upgrade model. And so then this is the Najib and Ha are the folks, remember, that collected the neural data to make this comparison even allowable. So they deserve a lot of credit for this, too. So remember, the idea is we're comparing brain features with machine features. And I think Rob already introduced this nicely. But for those of you who weren't here this morning, basically the idea is we'll take these object recognition 1.0 images that I'm telling you, HVM 1.0, the image set that we've been created. And so this is going to be our test. We're going to show these same images to both the neurons and to the model. And so we show it to the neurons. There's a pixel representation that goes through the brain, and it goes through the brain. And then there's some IT cortex representation and some IT state space that I've been talking about. And this is just a schematic, maybe cars and fruits laid out in this space in some way. And so this is the feature space of IT that I've been talking about. And you can take these same images, again, a pixel representation, through a deep neural network, and we're going to test a few of them. And then they have their representation, their output feature space here. And then, so we take the same images, and then we have referee at the end, an evaluation stage. In fact, it's 2 referees. 1 referee, we were doing linear SVMs, the kind of things I've been showing you throughout. Charles also did a kernel analysis, if those of you are interested in that. The results are very similar, so I'll just mostly focus on this. But we had 2 kinds of referees at the end. And again, so now you get the setup. So here's what basically happens. You have accuracy on this axis. Here's human performance on these kind of HVM tasks that I've been showing you. This, I want to point out, is performance during a single fixation sample. I'll show you that in a minute here. So the basic idea, I mean, Rob said this earlier, for those of you that had his talk, humans are not perfect. But remember, the images are, first of all, not masked. They were shown for 100 milliseconds. What we were trying to do, let me show you that here if I can get that to run. We were trying to emulate what I showed you earlier, that we don't take in the whole scene at once. You're going to be able to do core recognition by something like these glimpses that are basically only a couple hundred milliseconds as you explore the scene. That's what we were testing. So we asked how well humans did in those glimpse conditions, core recognition. So these images were shown for 100 milliseconds. We also did at 200 milliseconds. Their performance does not improve much between 100 and 200 milliseconds. So it just turns out that it's just sometimes hard at high variation or with to actually say, was that a chair or was that a table, right? So it's like there's some difficulty in doing that in 200 milliseconds. And so that's the performance of humans is shown there in the white line, and on the black, or in the black there you see the IT neurons. Sorry, I've lost all pointer ability. I'll just hand wave a bit. So the black that you see there is the IT cortex, and you see the extrapolation to 500 features of 50,000 neurons that I showed you earlier to match human performance. And you see the V4 down below. So we have both neural and human benchmarks there on our image set, and then we can now look at the machines. So a V1-like model, it's a V1-like, so you could call it neural, but it's actually a model, and it was down near chance performance. HMAX model was a little bit better. The Liadol model, this is work from Adam Eng's group, and it was still pretty low. But then we were quite impressed and surprised that Krzyzewski model really kind of blew this thing way up and is already up at near human levels and IT levels. And Rob's model is slightly better as well. So these models are really impressively high, at least by these kind of benchmarks. And again, I don't think they had no idea about our benchmarks. They didn't optimize on any of this, and I think this is a nice sort of fair test that we hadn't put together planning to test their models, but it turned out nicely that we were able to do that once these models became available. So now we're interested in the kind of questions that came up earlier of looking at these models and the way we've been looking at our own models to ask how well they explain neural data. And I maybe will skip through this as a kernel analysis method to look at if you vary complexity versus precision. For the machine learning folks in here, they may be especially interested in that. We could talk about it if folks want, but let me just sort of show you the results of that it gives you a very, very similar result. It actually is more dramatic that the only models you can see, if you read the plot there, I'm sorry I can't point, up near IT cortex extrapolation, the green and the red lines are the Ferguson and the Krzyzewski models. Those are both right around IT, and the other models and V4 is all down near the floor. And so high is good on this plot here, and it's sort of even at all complexity levels, is 1 over the regularization parameter. So that again just verifies what I already showed you, maybe even looks more striking than the last plot. I was worried. I said, well, look, this isn't fair. These models, they have thousands of features, and we only have 168 neurons. How can that be a fair game? So we did things like take the models and subsample them. And here we see number of features down on this axis, and we subsample down to low numbers of features. And so you can see those are random samples of both those models in green and red, and IT cortex subsamples of IT up to our max sample number shown in black. And you can see those curves kind of parallel each other, and IT is sort of sitting right between them. And based on extrapolations of the curves, and you can't go as high with IT, but we don't expect, even in the limits of max numbers of IT neurons for this thing to change at all. So this isn't due to the number of features that we think we're sampling, although, again, these are extrapolations. So the upshot is that the field now has at least 3 candidate hypotheses for the brain's ventral stream mechanisms. I mentioned HMO, there's supervision, and then there's the Zieler and Fergus model. Again, these models are all great improvements over what we had, but all clearly wrong in lots of ways, and that they're only explaining half the variance of the neurons. So glass half full, glass half empty, but a lot of progress. So this is not, we can't declare victory, but making good progress here. So any of you interested in this benchmark, it's publicly available. The images I've been showing you, if you have a model, you want to try it out, or you want to compare it to neurons, go to this site or talk to me afterwards, and you can find it there, as well as a link to the paper. Okay, again, I already said this, but it's too early to declare victory with these kind of models. This HVM, we call this object recognition 1.0 for a reason. We knew this was just the first step of testing object recognition in a quantitative way in humans and monkeys and machines. So can this satisfy more stringent predictions? Even on the images we have, there are stronger tests that we can use than we've applied here so far to ask about the image-by-image patterns of confusion that came up in some of the questions. And really going forward, what we really want to do is test other object recognition challenges, things like occlusion, multiple objects, illumination. So that's what we're thinking about is what we'll probably imagine for 2.0, and that's what we're working on now. We have to redo all the human data. We're collecting more neural data, but so now there's going to be another turn of the crank here along these same directions. So I think that I've given you a feel for what the comparison there between machines and monkey neurons and human behavior. And I think one thing to take from that is it demonstrates that the recent bio-inspired models really starting to rival the brain in terms of object recognition performance. So let me stop with that part there and maybe take questions on that. But maybe since it's the last section, I'll just want to say a few concluding remarks or take-homes. For those of you who aren't paying attention or fell asleep, here are the things to maybe remember. Invariance is the crux problem. Many of you know that, but you need to keep that in mind. If you don't, simple IT population rate codes are sufficient so far to account for unfettered human object recognition performance, at least on this 1.0 kind of testing. We're now testing monkey behavior and sharpening the behavioral test. Again, I mentioned 2.0. These IT codes are computed reflexively. Again, the monkey is just passively viewing in about 100 milliseconds and are likely shared by humans and non-human primates. We believe that not only from our comparisons of monkeys and humans on behavior, but from work like Nico's work that I showed you comparing monkeys and humans at the level of fMRI, voxels, and IT neurons. The key transformations likely live between V1 and IT. We know a lot about V1, not everything, but we think we have pretty decent models of V1, and now we're starting to have much better models of what's going on up to IT. We've been searching that model class of bioconstrained models. High-performing models can accurately predict IT neural responses, at least 50% of the variance, and their intermediate layers predict V4 responses. And as you saw at the population metrics, even stronger improvements. Other artificial deep convolutional networks are now rivaling neural and human performance on our benchmarks. Those are now viable hypotheses for ventral stream mechanisms. And the really one big take-home message is that the brain really has guided a lot of computer vision. A lot of these successful models that you see today, convolutional nets, that's brain-inspired, LN units, brain-inspired supervision. A lot of this came out of looking at the brain and getting inspiration from that. And now I think things are starting to turn the other way, where now the models are actually starting to perform well, and they are now becoming hypotheses and providing ways to get a deeper understanding of what's going on in the brain. And I think I've given you some examples of that, as now we're being able to start to predict the responses of neurons in stronger ways than we had been able to do before. And I think that's a nice synergy between neuroscience and computer vision. And hopefully there'll be another round as we think from neuroscience will help inspire next generation of computer vision models. I really want to thank the folks in my lab who did a lot of what I presented today. The folks in red here, Charles Cadeau, Ha Hong, Dan Yeamans, Najib Majaj, Nicolas Pinto, Ethan Solomon, Dave Cox. Those are especially folks who contributed a lot of what I showed you today, but everybody on this top contributed something, and especially thank the funding agencies there. And I really want to thank all of you for your time, those of you who stayed here for 2 hours. And I think we have at least 10 minutes for questions that I've preserved here. So again, thank you. Do you want? Yeah, sure.
So these models obviously are inspired, and you put them together through a sort of engineered approach, but then you go on and you optimize them further. And so I'm wondering if there might be some insight into how the brain is functioning by sort of cracking open the black box of what the optimization is improving about those models. And have you looked at that at all?
So you mean, I'm not sure if I'm following that, what you might do. So doing some changes in the optimization techniques and seeing if that leads to different, better fits of the neurons, for instance, something like that?
I'm just wondering if the parameters of the optimization, if you looked at them closely, are insightful in a way to understand how that system is working and therefore to infer
the parameters of the optimization.
where they ended up.
The final architectural parameters. Yeah. Yes. Oh, okay. So, right. So one interesting thing to say there is it's one very reliable thing that comes out of this optimization that Dan likes to point out is there's lots of what you might call face units that come out of the thing. And, that's something that we see often in IT that I alluded to. So what's sort of striking is that those come out of this kind of approach not because we're telling the system that, hey, faces are the thing you better succeed on or you're going to die as a machine. We're just saying it's one of many things you have to solve. And it's in the shape space, it turns out that, hey, those shapes are close together. So you probably need to optimize a model to deal with the nearby similarity of those shapes. That's what's going on in that case, I believe. So Dan may elaborate more on that, either here or in his poster if you like. But I mean, there are things like that come out in the architectural space. Dan also has, and he could talk about this in his poster, an analysis of the architectural parameters and which ones are most important and which are not. And I think maybe that would probably be, I would point you there to start to go after those questions. Yeah. I said Nico, yeah.
This is fascinating stuff. So computational neuroscience has struggled explaining variance in single neuron responses to arbitrary images for a while, right? So in V1, I think like more than 10 years ago, if I remember correctly, Bruno Olshausen estimated 30% or something, I don't remember the number. So then V2 is harder, V4 very hard, IT
don't try.
Was thought of as impossible, right?
Right.
So now you're saying 50%, that's a very high number. Is that really to arbitrary stimuli, or can you comment on that?
So it is to arbitrary stimuli within that class. So of those kind of, you saw how we generated the images, right? So that, if you say, well, look, first of all, there's no color. There's things like that. I mean, but generally there was a central object in them. But those were pretty rich stimuli. So it's arbitrary within that class. We think it would generalize to a lot of things, but we haven't tested that. But I want to make a point about the explained variance number and what you're saying about Bruno and Jack. So there is an important point to be made here. We are just fitting the mean response in that 100 millisecond window out of each IT neuron. So each IT neuron gets one value for each image, and that's what we're fitting. And we're explaining 50% of that. Often in the studies of especially V1, people look at more fine time resolution, and they want to predict even a 10 millisecond bin. And we're kind of saying, look, we went 100 millisecond average. So there's a difference in sort of goal there, right? Now, our goal, I would argue, is motivated by the behavioral studies that I showed you. Is like that time window is what we need to predict behavior. So that's the thing we want to explain. But that doesn't mean that, if we start looking at the narrow time windows, we'll explain variance numbers will probably go down. Whether that's important or not for understanding the brain will maybe be still an open question. But that's what I want to kind of make that sort of clear point, that just because we're saying 50, if we do V1 with 100 millisecond time windows, I don't know what the number might be. So there's not the game being played is a little different in the different areas. And I was just at a talk with Bruno yesterday, and they were still saying, look, it's about 10%. But they're actually trying to explain trial by trial variance, not even average trial 10 millisecond. Now they want to explain every trial. And again, we're averaging over trials. We're assuming that's variance we're not trying to explain. So we have to first be clear as a field of which fraction of variance is important before we kind of get serious about comparing that. So I just want to say that the 50% variance is a huge gain over zero in effect, right? But compared to V1, you have to be a little careful there. But even Jack's models, they tend to be a little closer to what we do. And their earlier numbers were more like 20% in V4. I think they've improved more recently. In fact, using some of these models, I saw presented SFN, where they're using some of these models to try to fit V4 and have made some gains there. So I think the answer is it's a little, I'm not giving you a clear answer, except to say that I think this will now start to force us to lay this out more clearly. And I think with your history, we'll start to, maybe we can do this together. And I'm sorry, I didn't even know I didn't put all that stuff about your work in there knowing you were going to be here, but now it's nice that you were here. So if anybody's interested in the stuff I presented on Nico's work, there he is.
So I think you might have just answered my question. So I don't know if you were asking, but you sort of started off by saying, rate codes are this sort of, really weak guess at what might be coding, but you do have explainable variation in your data. So once you've predicted the class object, you still have all of the geometric data that you control. So have you tried to just take the residuals of those regressions and just say, can I predict the spatial orientation? Maybe the rate code is actually predicting more.
Oh, yes. Yes, it does, and it does do that. And we have done that. I just didn't present it here. So I sort of alluding to in the last session, there's kind of I'm kind of echoing a neuroscience view. There's a view that, like, oh, the codes we're using have got to be too simple. And there's that sort of argument to be had, and was that. But then you're making the other argument, which is like, well, look, maybe the rate codes actually have a lot of power, and you've only told me about part of their power. And in fact, when you do use those rate codes to estimate things like position, then we can get very good accurate estimates of position out of these things as well. And we just submitted an abstract to cosine on that. But this is pretty cool because in those images, you can take the position of the object, and you might say, oh, V1 could do that. But in those cluttered scenes, V1 can't do that. So you actually, there's information there about position and pose and scale. We're now trying to benchmark that against human abilities. That's why I haven't really presented that, because everybody, the guys show me, hey, look, we get these good predictions out. And I'm like, well, what's my reference? I need to put it against a human to give it some context, just to say there's information there. It's there. How much and how quantitative perfect it is still, and does it force us to change the rate code or change to a different code is what we're doing next. But that's a great point. And if you'd like to, again, go to Dan and Ha's poster, and they can tell you more about what they've predicted so far, or I can talk after some more. Yes?
Do you think things would change a lot under previewing, or do you think that your sort of setup of flashing images sufficiently mimics previewing that you'll see?
So right, so I kind of alluded to those samples. I took those samples out of the image. I said, well, instead of actually having the monkey sample it, I sort of forced the samples down the monkey's throat, right? And in fact, they weren't even from the same scene, but I made arguments that you don't need the scene. But as a postdoc, I did work on exactly that question. So I had free viewing monkeys, and then I replayed for them identically to their retina the same images. And they weren't natural images. They were more simple than this. And I was recording in IT, and the responses of IT units are nearly identical under those two situations. So that's why I don't worry too much about that. There are advantages to free viewing because the monkeys like to do it. They don't like to fixate. But then your disadvantage is you've got to be careful about your stimulus control and the things I had to worry about as a postdoc. But it's a great question, but that's what we know so far. There may be important things that have to do more with context and attentional effects, but just eye movements per se probably have very little effect on at least IT neurons based on that earlier work.
So maybe you answered this earlier in the talk, but I was wondering whether any of the sort of stages in like V2 or V4, for example, did you have any evidence to see that they corresponded to actually rotating or rescaling some of the input images, like actually generating variance that, like if you saw a car in a hill, it might rotate it to a horizontal car so that I can classify it?
Well, the way we think of these networks working, and I think the same, Rob may want to comment on this, you have these convolutional deep networks. You don't actually sort of rotate the object anywhere. You just build features that have correctly tuned themselves that they're going to be tolerant to that rotation, or at least project it in the right way. And it's going to do that gradually through the system. So you won't go into V2 and find some rotator circuit or of that nature. So I don't even know what we look for in neurons to sort of ask if it's rotating, right? I mean, what we can say with models on V1 and V2 is just to predict the performance just gradually gets better in terms, and then it reaches human levels at IT, and the pattern of performance gets more matched to humans as you go up. But you're asking more about the detailed features of what are these V2 units doing, and the closest we have to that is what I showed you on V4 and IT fits from the model, but I haven't given you what humans are often looking for as something more than just saying you have a model that explains variance. You want me to say, well, what exactly is going on? And we like to try to come down to something more precise than that. And this is, I gave Rob a hard time at our recent meeting we were at about, okay, you now have a model. I'd say the same thing to Dan is like, can you take that model apart and understand it in a way that's sort of more human interpretable? And that's a challenge in itself, I think, not only for neuroscience, but for folks who have this in silico and can show it as many images as they want. And Rob's done some nice work, if you read his tutorial this morning, on trying to do those kind of things. So I don't know if you want to say anything more on that yourself. No? Okay.
You did say more isomorphic early on and then more abstract.
Well, right, so isomorphic early on is that, but I wasn't talking about temporal rotation or anything, right? It's just a feature vector that's changed here. But I'm only showing static images. If we were moving in time, we'd expect the vector to also be moving in time. But we don't think it takes the feature space and then computes something and then re-rotates it and then projects it up. It's just dot products in effect with nonlinear areas. That's what these models are. Yeah. Any more questions? Yeah.
Can you comment on the suitability of the RSVP paradigm for comparing models and humans? Obviously, models don't have any idea about time and presentation time, and that's probably why you get a better performance of Wheeler and Fergus over humans. And if you increase the presentation time 50 more milliseconds, probably main pattern will change.
So I think I said that you're saying the question is about the so you said RSVP, but when we do the studies for the human, we present an image for a short duration and then they get a response screen and they give an answer. So they're not seeing a sequence of images. I just show you that to give you the intuition that you don't need a long period of time. So we don't present, the RSVP only comes when we test the neurons. So it's a passive monkey, and there it's just to get as much data out of the neurons as we can. So for the behavior of the monkey or the human, we show an image for 100 milliseconds and then a choice is made. We've also done it at 50 milliseconds and 200 milliseconds. You start to drop off at 50, you don't get much better at 200. You need to get to longer time windows, more like 500, or you start making eye movements, but you start to see improvements. So now we're in a regime where we're going to start taking into account eye movements. And again, our focus was on core recognition, what happens in that first few hundred milliseconds. So we don't think the performance numbers are going to change that much. In fact, we know they're not going to change that much for going for just another 100 milliseconds. But you go to a second, as soon as you start opening it up, now you have eye movement. So now you've changed the problem, and a lot of other things have to be considered to do that. So we kind of redefining what we mean by object recognition. That's 3.0 or 4.0. And I'm not saying we shouldn't go there. It's just we want to kind of gradually build our way up to that. So again, don't take this as the limits of all humans can do. But now we kind of sort of have a foothold to sort of now we can extend it and build on it. Okay, so again, I try to say at the end, we're not declaring victory. Models have not declared victory. We haven't declared victory of everything the ventral stream does. There's a lot of progress here, but the game is not done. There's far more to do. As I alluded to at the end, more tasks, longer times, attention, all of those things have to be layered on. As we call it core, we need to understand the core modules first, and then we can, I think, have a deeper insight as to how those things might get layered on. I mean, my dumb model right now is you have somebody directing eye movement, somebody like the dorsal stream. You have an engine digesting, and then you have semantic readouts at the back end. That's the simple model. Now, of course, you have some knowledge web behind there and all that, but that dumb model may be enough to explain a lot. You just have to direct eye movements, tell me what it is, tell me what it is, tell me what it is. How much these contextual effects that are going to be there, not just directing your movements, but sort of giving you some guesses about the object before you make the eye movement. Those are questions that are of interest. But
how about going this direction, comparing deep learning models and human IT cortex, human vector stream? Do you think this is the right way, or maybe this ends up to learning about ventral stream, as opposed to just focus on ventral stream and try to figure out what is the algorithm, just putting them in contrast?
Oh yeah, I mean, it's right. I hope I didn't come up here and say I'm an advertisement for deep neural networks, other than that's what the brain is in some sense, right? So depending how you define it, right, there's multiple levels and it's a neural network, right? But I didn't say, oh, what we have to do is all go do what Alex is doing or what Rob's lab is doing. I'm saying those are models that are now hypotheses that neuroscientists should pay some attention to, because look, they're kind of in the game. But that doesn't mean that we aren't going to continue to do this kind of neuroscience work, that a lot of which I didn't tell you about today, of trying to understand the ventral stream. So I was partly highlighting that because of this audience, right? That's why I built this talk this way. But there's a lot of work to be done on just going after the more classic ways of trying to get at this problem. But when it comes to explaining neural response variants, I think there's a lot of value to be gained there. But when it comes to questions like learning and things, there's a lot more experiments to be done just on the neuroscience side, some of the kind of questions that came up earlier. So it's a combination of both that's needed to make progress on understanding the ventral stream. We can't, but neuroscientists that do not pay attention to the networks that are being produced, I think, are going to do so at their own peril. That would be what I bet you Nico would agree with that. So I don't know. And I think we're out of time. We should probably end. Yeah.
Okay, well, let's thank Jim again for a great tutorial.
Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.